Bismillahirrahmanirrahim. Assalamu alaikum everyone. Welcome to the stream of knowledge. Today we will learn about peptidoglycan. In our previous lectures, we have studied that bacterial cell wall is made up of peptidoglycan. We also learned that in gram-positive bacteria, this peptidoglycan layer is thick, while in gram-negative, it is thin. In gram-positive, the cell wall of gram-positive bacteria is made up of only peptidoglycan layer, while in gram-negative, in addition to the peptidoglycan, there is outer membrane or lipopolysaccharide layer also present. Today, we will learn about what is this peptidoglycan that made the bacterial structure and what is the structure of this peptidoglycan and what is its function. So let's start without wasting our time. As the name suggests, peptidoglycan, there are two parts of this name. Peptido and glycan. Peptido, it looks like a peptide, and peptides are present in proteins. So the term peptidoglycan is derived from peptides and sugar or glycan that make up the molecule. Peptidoglycan is also called as murine or mucopeptide. This structure is 2D, while the real peptidoglycan cover the bacteria or peptidoglycan surround the entire cell in 3D structure. One thing you should keep in mind that peptidoglycan is only present in bacterial cell walls. Now we will discuss about the function, then we will move towards the structure of peptidoglycan. As we know that the peptidoglycan form the cell wall, so as we already know that cell wall in plants also provide the support, therefore the just like that in bacteria the peptidoglycan or cell wall provides the rigid support. It also maintains the characteristic shape, as we already learned in our previous lectures that the characteristic shape, cocci, bacilli, and spiral shaped is maintained or due to the peptidoglycan or cell wall of bacteria. One more important function of peptidoglycan is it protects against the osmotic pressure. This point is important, so learn it carefully. For example, this is the bacterial cell, and inside the bacteria there is high concentration of salt, while in the environment there is low relatively to the bacteria. So we know that due to osmosis, water move towards the cell of the bacteria, and hence the bacterial cell can prone to burst, but it does not. Why? Because peptidoglycan protect it against this osmotic pressure. In this lecture we also studied that peptidoglycan is the main site of some antibiotics action, for example penicillin, and this peptidoglycan is also degraded by lysozyme. We will learn these 2 points after some time, after a few minutes. Now move to the structure of peptidoglycan. As you can see, this is backbone. This backbone is glycan part, and this part is the protein part. The carbohydrate backbone is made up of N-acetylmuramic acid and N-acetylglucosamine. This is N-acetylglucosamine, and these are N-acetylmuramic acid and its mufoma chain. This is called a glycan chain. N-acetylglycosamine or NAG attached to NAM and NAM attached to NAG, and it form a long chain. Then with this NAM, a tetrapeptide structure is attached. As you can also see in this figure that these are alternating N-acetylmuramic acid or NAM and NAG molecules. These are alternating, and this layer and this layer, then both these layers are connected. But how? This is a tetrapeptide with NAM. The tetrapeptide is attached, and these two tetrapeptides are cross-linked by a peptide interbridge. This is peptide interbridge between two tetrapeptides. So we have learned that the sugar or carbohydrate backbone, which is made up of alternating NAM and NAG, are connected with each other by tetrapeptides that are further cross-links. In this way, the two backbone chains are connected and form a interwoven network. that surround the entire cell. As I previously told that this peptidoglycan is a main site for some antibiotic action, for example penicillin. But how? These two tetrapeptides are cross-linked by an enzyme transpeptidase. So, penicillin, cephalosporins, and vancomycin-like drugs inhibit the synthesis of peptidoglycan by inhibiting this transpeptidase that make this cross link between the two transpeptides. So, if this transpeptide is not present, or if this crosslink is not present, the structure will be the broken, means these two backbone will not connect it to each other. Then what happen? The water will enter to the cell and cell will burst it. Now, what is the action of lysozyme? Lysozyme, which is present in our tears, mucus, and saliva, break this glycoside bonds between NAM and NAC, and hence bacterial structure is disturbed. That was all about the peptidoglycan. Thank you for watching. Do subscribe, like, comment, and share. Please, guys, subscribe. It will motivate me. Thank you.